Hey guys, one of the comments I got last time was that I mentioned you might get cat pictures and then there were no cat pictures. I'm sorry about that. Here's a small cat picture. This is one of my three cats. This is Nadia. Oh, she goes. She's our elder female cat. She's a little terrorized by our younger male cat, Oliver. But having the three of them around is definitely a help at times like this. I'm going to take myself off the screen. I assume I'm a distraction rather than a help. If that's wrong, if you'd really like to see me, minimize or something in the corner of the screen, I can certainly do that. I can make myself a little box in the corner. And if it helps to see my lips moving or whatever, then let me know. More than happy to do that. But for the time being, I'm going to take myself off the screen. Here I go. All right, so what are you seeing? What you're seeing on the screen are neurons from an Alzheimer's brain. And beautiful as they are, that buildup of gold is actually marking the accumulation of a protein called phospho-tau. That's an abnormal form of the protein tau. It is a cell cytoskeleton protein that helps build the cell wall, cell structure. And when it's abnormally phosphorylated, what happens is the cells physically shrink. And so what you see is that the synapses get lost because the cells are shrinking in on themselves. And so you get this loss of cellular connectivity and atrophy of the cells. It's a key feature of the Alzheimer's brain. I put this slide in here as a reminder. I've sent a couple of emails about how this is going to work. Definitely class presentations, still part of the grade. I mentioned, Rachel and Jake are going to be our guinea pigs. And if you guys can try to get the best you can do, whatever that is, to me as soon as possible, that will really help to give some context. I'm using Zoom to make this slideshow video. You guys can have a private Zoom session where you do the same thing, basically share the screen and both talk, record that, send me the link. I think that should work fairly well. If there's a problem, let me know. Obviously, I'm not going to hold you to the original due date. Life has been completely weird. But if you can try and get me a video presentation from you guys, sometime this coming week, please. So sometime in the week of March 23rd, that would be great. And it will give some context and an example for everyone else. I will put the slideshow that you sent me up onto Blackboard as a PowerPoint file, but a video will be really helpful. Thanks. If that's a problem, let me know. We'll work with it. All right, onto content. So last time, we were talking about homeostasis and the concept of a set point, a value for something, whether it's water content, blood glucose, temperature, whatever it is that your body wants to maintain. When if you get too high or too low away from that point, there are mechanisms in place to sense that and to correct it. So for thirst, for instance, there are detectors that will say, hey, there's not enough water here. There's too little concentration, there's too much concentration, there's too little volume, you've lost some blood or whatever. Go fix that. The primary mechanism for fixing it is to drink, take some water in. And then there's a feedback loop that says, okay, we've sensed water is on the way, you can stop now. And I mentioned last time, that feedback loop for any of these homeostatic variables, whether it's temperature or water, whatever it is, that kicks in before, in fact, the brain would see the effects. So for thirst, before the water ever gets to the brain and changes the concentration, you've got sensors in the mouth, you've got sensors in the blood vessels to say, okay, some water's on the way, thanks, you can stop drinking now. Otherwise, you just keep overshooting like a poorly controlled thermostat. One of the ways that happens is that you have these specialized osmometric neurons, and when the concentration of water outside goes down, water flows out of these cells. They physically shrink, and that causes them to change their firing rate. That's a direct physical signal of, hey, we're thirsty. The sensory organs for thirst, I mentioned, are located right around the third ventricle, so they're really on the very outside of the brain. That allows them to be more rapidly able to sense changes in the concentration of fluid, in this case in the CSF. And the summary slide for drinking is really here. You've got sensors for osmometric thirst, for concentration, for volumetric thirst, how much total fluid do you have? Are you bleeding out? That's going to make you thirsty, oddly enough. And then all that information is integrated in the medium preoptic, which regulates this drinking response. That's pretty much it for thirst, but it's a really excellent example of homeostasis and a set point. Hunger is a second good example. You are definitely trying to maintain a certain level of weight, of hunger. Body mass, as many of us will know, myself certainly included, as your body mass increases, it's actually really hard to go back the other direction. We've evolved for thousands of years under conditions of food scarcity. And so this is another key concept, that you have adaptation of the set point. When something changes long term, So as you gain some weight and stay at a heavier weight, your brain says, huh, okay, that's normal. That should be my new set point. And all those homeostatic mechanisms kick in when you try to move away from that set point. So you gain 10 pounds really quite easily. And then your brain says, great, we're a little heavier. We've got a few more fuel reserves. I'm going to defend that point. And so your brain kicks in mechanisms to maintain you at this new adapted set point. We'll see it in a little later in the class. That can be true for things like blood glucose too. So if you are chronically diabetic and you have lots of very high blood sugar, your brain will stop responding to that. That can be true for hypoglycemia, low blood sugar. We'll come back to that. You know, as I mentioned that obesity, weight gain, is not an individual phenomenon, it's a mass phenomenon. There are millions, probably around the planet now, billions of people that are obese. These mechanisms that have evolved to regulate hunger don't necessarily always lead us to the right outcome. One of the main reasons we talked about that obesity has increased really so rapidly over the past 30, 40, maybe 50 years now, is this increase in a particular form of sweetener in calories, fructose. Fructose is very commonly used in most processed goods, usually in the form of high fructose corn syrup. And one of the key things about that is that although glucose gets metabolized in this glycolytic pathway through fructose 6-phosphate, in fact, that whole step is regulated by the enzyme phosphofructokinase. And the key point there is that phosphofructokinase is turned off by ATP. When you have lots of ATP, so your body is in a high energy state, you don't need any more fuel, then glucose doesn't get metabolized. So you don't build up any more fuel. In contrast, fructose itself doesn't go through that step. Fructose goes via fructokinase to fructose 1-phosphate and so on down. The key point though is it skips this regulatory step. There is no feedback loop to allow ATP to turn off this metabolism. So if you absorb fructose into your gut, you're going to metabolize it, you're going to store it, and basically you're going to store it very effectively as fat. And so what you see is that taking in fructose sweetened drinks is very strongly associated with gaining weight, and in particular gaining fat mass. I mentioned homeostasis. I'm going to come back to that several times. Here's another really good example. So homeostasis for blood sugar. You have a certain level of blood sugar that your body thinks is good, that it wants to keep and maintain. When that gets too high, when you have too much glucose, your body starts to release insulin from the pancreas, causes glucose to go out of the blood and stored as glycogen. Conversely, when blood glucose is too low, glucagon kicks in, puts some of that glucose back into the bloodstream. So it's a homeostatic cycle. Insulin's acting, we went through this last time, via the signaling cascade I don't care about. Actually, I care about it a lot. It's what my lab works on. I don't care that you care about it. And then what happens as an end result is that this glucose transporter, GLUT4, goes up to the cell surface, allows more glucose to come into the cells. So you've got an increase in glucose outside in the blood that causes the body to release insulin. Insulin then binds to these insulin receptors, allowing glucose to get pumped into muscle cells, fat cells, and taken into the system. We mentioned that there are two centers, both in the hypothalamus, for hunger and for satiety. And in particular, those centers contain these glucose responsive neurons. So GE, glucose excited. These spikes, these endogenous action potentials, are very high when there's a relatively high level of glucose around. But if you move that to a low glucose state, the neuron fairly quickly shuts off, stops firing. You bring back high glucose, it starts firing again, and so on. This is only glucose. If you put fructose here, the cell wouldn't respond. If you dropped some olive oil onto it, the cell wouldn't respond. These cells are purely responding to glucose because that's the brain's fuel. And the brain in this regard is pretty selfish. It only cares about the fuel that it's going to use. Table sugar, remember, is 50-50. It's a disaccharide. It's half glucose, half fructose. And that fructose is treated very differently to glucose. Doesn't get to the brain, just can't get through the blood-brain barrier. Goes through different glucose transporters. In particular, GLUT5 is in the gut, is in the intestine. And so it's very effectively taken in as a fuel, stored in fat, but doesn't stop you feeling hungry. If you drink a whole ton of fructose, I don't know, you take a Coke, for instance, then the fructose in that drink is not going to stop you feeling hungry. The reason for that is your brain doesn't notice. The fructose doesn't get to the brain doesn't respond, it doesn't turn off those hunger mechanisms. Likewise, doesn't trigger insulin release. So only glucose triggers the release of insulin to regulate that homeostasis. We went through the molecular mechanisms of how GE neurons sense this glucose and respond. The key enzyme I mentioned is the glucokinase. So the very first step in the metabolism of glucose is it has to go to glucose 1-phosphate, as it happens, but it's metabolized in most cells in the brain, in most neurons, by hexokinase. Hexokinase is basically maxed out the whole time. It's maxed out at very low levels of glucose, so it doesn't respond to changes in glucose. It's always just maxed out. Glucokinase does exactly the same job, but it's really only found in these specialized glucose-sensing neurons because it changes activity in the range of glucose that these cells actually see. And so when you get more glucose outside these cells, because they're using glucokinase, they're going to make more ATP. As they do that, they directly bind to a potassium channel, they close the potassium channel, cell depolarizes. So to take you through that, as you get more glucose, you increase the amount of ATP being made, you close this particular channel on the cell surface. So this GE neuron, you're closing this potassium channel. Potassium can't leak out, cell becomes a little more positive on the inside, that's depolarized, and so the cell is going to fire more easily when there's glucose around. It's glucose excited. And you might recall I mentioned the difference with a GI neuron, a glucose inhibited neuron, is that you still have this response to ATP, but in this case, it's affecting a chloride channel. And so chloride is being kept inside the cell. There's not very much there to begin with, but what there is being kept inside. And so what you see is the cell hyperpolarizes. It actually gets more negative on the inside and fires less. So glucose inhibited. You might remember this is the cystic fibrosis transporter. It regulates salt movement into all kinds of things, mucus, sweat, salt, and so on. That homeostasis for blood glucose level is basically regulated at the level of the pancreas. So the pancreas is the organ that releases both insulin and also glucagon. It's actually insulin from the beta cells and glucagon from the alpha cells, the two cell types in the pancreas. And so here's the homeostatic cycle. If your blood sugar is too high, the pancreas releases some insulin and tells the liver, hey, take up the glucose, store it as glycogen. Tell it to muscle cells, take up that glucose, store it as glycogen. So now you have less glucose. So far, so good. If you go a little too low, you've got low blood sugar, this is hypoglycemia. Instead, the pancreas releases glucagon and tells the liver, hey, we need some more glucose. Break down some of that glycogen, pump it back into the bloodstream. So you've got a homeostatic system, two actors, two hormones opposing one another to regulate blood glucose. I also mentioned that although the brain is very selfish, other parts of the body do talk to it. So the liver, the gut, and so on, they all send signals, not just for glucose, saying, yeah, hey, I've eaten 6 pounds of cheese, you really shouldn't be hungry anymore. And all of those signals, both long-term, adiposity, what's your overall long-term fat state, and short-term, satiety, have you recently eaten? All of those signals get taken up and eventually synthesized at the level of the hypothalamus. We generally think of the arcuate nucleus, the ARC here, the arcuate nucleus of the hypothalamus as being the key central regulatory point for all these hunger and satiety signals. We mentioned also, there's not only hunger as a reason to eat, there's also pleasure. You eat something that tastes good, it makes you happy. One thing that we see is that the level of dopamine release, so the chemical marker for pleasure, dopamine being released in this medial forebrain bundle. The next topic we're going to cover in class is drug addiction. We'll talk about that dopamine system a whole bunch more. But for now, look at this top slide. Brain metabolism, roughly the same in the brain of control humans and really obese humans. But this dopamine binding, much, much less in the obese brain. And so basically the shtick here is that for any given amount of pleasurable food, obese individuals may be getting less pleasure. And so they need to eat more food to get that same amount of pleasure. You can think of this as being another example of homeostasis. Humans want to have a certain amount of enjoyment. And if that means you need to eat 3 donuts, okay, 3 donuts it is, leading to obesity. I showed you this graph last time, this correlation between increased BMI, so greater fat mass, and reduction in dopamine binding. The fatter you are, the less pleasure you're likely to get from a particular stimulus. Just like last time, this will be a point where I really ask for questions. If you have one, jot it down, send me an email, put it in the Blackboard chat, do something to let me know that you have a question. We'll obviously talk synchronously with most of you, I hope, in the class slot, but don't let yourself forget the question. Write it down somehow, reach out to Corey, reach out to me, make sure you get an answer to. This is the first, of the non-me class papers that have been reviewed. So I said we'll get the video up from Rachel and I think it's Jake as soon as possible. The methods here are really pretty cool. This paper is 12 years old now, and the methods are almost cutting edge even still. I really like them. I really like the logic to the paper. That's why I picked it for you guys to read. It is pretty likely I'm going to quiz you about it at some point because I really think it's a cool and very clean example. So here's the background. There's this class of neurons, AGRP neurons, and they release three things. So we've talked about how each class of neurons only releases one neurotransmitter. That's basically true. So in this case, the neurotransmitter is GABA. They're also releasing, though, at the same time, these two neuropeptides, these two other things that can modulate target cells. So these cells actually release three things at the same time. First sentence, they're important in regulating body weight. If you take out those neurons, this is previous work, then you change feeding behavior, you change body weight. So we know that this small set of neurons, they're in the hypothalamus, they express MPY, they express AGRP. We know they are important for regulation of feeding, because if you lesion them, you get problems. However, if you take out two of those three things, if you have animals where you've deleted AGRP or animals where you've deleted NPY, not much happens. So again, here's the previous work. The key part is highlighted down here. AGRP negative, so knockout mice that don't make AGRP, NPY negative mice suffer no obvious feeding or body weight deficits and maintain a normal response to starvation. So these are mice that don't have either of those two peptides. Those peptides simply don't exist, and yet they look pretty cool. So you've got these cells that we know are important, and they release three things. Two of the things don't seem to matter. Obvious conclusion, maybe it's GABA. Which is great, except that it's really hard to get rid of GABA. And if you think about that for a second, you'll probably realize why. You can't just knock out GABA in the body. You can't even just knock out GABA in the brain. GABA is the brain's major inhibitory neurotransmitter. If you try to knock out GABA, it'll be lethal. The animals will simply have constant seizures from overexcitability. So it is not possible to generate a GABA knockout mouse. That would be a lethal condition. They wouldn't survive anywhere close to childbirth. That's kind of what they're talking about here. Because of the lack of methodological approaches, the physiological effect of GABA in the hypothalamus is unknown. Right. We haven't been able, before this, to manipulate GABA because it has these whole brain, really major effects. That's the cool part about the methods. Probably many of you found it a struggle to understand the methods in this paper. It doesn't surprise me. I'm going to go through it in great detail. I would like you to get it. And so if it still doesn't make sense after I've been through it today, maybe reviewed it next time, please let me know. Again, I'm going to be saying this a lot, right? I miss you guys. I'm sorry I can't do this in person, but don't feel that I'm not available for questions via whatever communication mechanism you feel, carrier pigeons might be a little tricky, but get in touch somehow and we'll work it out. Anyway, the basic method is they're going to do what's called flocks. So flocks is this method that uses two key parts, CRE and LOX. CRE, CRE, is effectively a molecular scissors tool. It cuts DNA. And it cuts DNA only and very specifically at a sequence called LOX. So you insert this artificial bit of DNA into the genome, and you do that on both sides of the bit of DNA that you want to cut out. Then, if you also have Cre around, if you have this enzyme, it'll cut the DNA, it'll get rid of that bit in the middle, and so what will happen is the gene no longer is functional because you don't have this bit of DNA in the middle, you can't make the functional protein. That whole concept, that whole system is called floxing. I don't know why it's called floxing, it just is. But if you hear someone say, hey, we floxed the gene, it means they put it under control of this Cre-lox system so they could delete it. And so now what you get is a system where, that gene doesn't work. In this paper, the gene that they floxed, the gene that they were able to knock out, is called VGAT. And as you can see at the top here, that's the vesicular GABA transporter. That's the protein that's responsible for getting GABA into the vesicles in the neuron. And so if this protein isn't there, GABA can't go into vesicles, it can't be released. Effectively, you turn off GABA function. All right. Last bit of this really important system. This is what it does. This is Cre-lox. So here's the Cre, the molecular scissors. These gray triangles are the lock sequences. Cre snips, you get rid of this bit of blue-orange DNA. Just either throw it away or turn it around in both cases. The last really key part of this is this whole system has to be under the control of a cell type specific promoter. So any of you who've taken any kind of genetics, the promoter, you'll remember, is the bit of DNA that tells the body which cells this gene should be turned on in. So if it's a retinol promoter, then all the genes you need in retinal cells get turned on. So you have a cell type specific promoter that controls expression of Cre. What does that mean? It means that only cells of this one type will have Cre activated. Only cells that are the particular type you're looking at. In the case of this paper, AGRP cells. So they used an AGRP promoter to control the expression of Cre. I'm going to go back up a couple of slides, back up to here. And so what you can see is that we have a neuron-specific manner generating these VGAT flox mice. And here on the bottom is the other key part. It's expressing Cre, right? It's causing these cells to make Cre only in the AGRP cells. So only neurons that are AGRP type neurons will be able to make Cre because the promoter that these guys picked is an AGRP promoter. So only in those small set of cells will this whole system get turned on. So you make two types of mice. These are two parent mice. In one type, you've got this Cre under the control of an AGRP promoter. And this mouse is entirely wild type, right? It's making the molecular scissors in some cells, but there's nothing for it to cut. So nothing happens. It's perfectly normal. The other target mouse is where you have these LOXP sites, where you have this floxed gene. So you put these LOXP sites into the VGAT gene. Again, this animal, completely normal. There's a gene that has these LOXP sites, but there's no Cre. So it's not going to get cut. Nothing's going to happen. Completely normal mouse. You breed those two together. Then you check. And some of your offspring mice are going to be the type that you want. And they're going to have both of these genes in their genome. So the target gene is floxed, and Cre is there in these very specific cell types. So in most cell types in the body, even in this offspring mouse, nothing happens. The gene is there for this target gene, but there's no Cre because it's not turned on in those cell types, so nothing happens. Only in the one specific cell type, in this case the AGRP cells, Cre is there, it cuts out this target gene, you get an effect. So think about what that means for our particular set of cells in this study. We've got the Cre mouse under the control of a particular promoter, the AGRP promoter. The gene that's being targeted is a GABA gene. It only makes a difference if the cell is packaging and releasing GABA. So when these two mice are bred together, here you have this Cre-LOXP mouse, and in some small set of cells, in AGRP cells, this gene's going to be cut out. And actually what often happens is the genome is constructed so you put what's called a reporter gene, a green fluorescent protein. So when you take out the target gene, instead the cells start fluorescing green to tell you, hey, your experiment worked. So in some cells, in the AGRP cells, that GABA gene has been taken out. If the AGRP cells are not GABA cells, if it's some other kind of GABA AGRP cell, then nothing matters. You know, you've taken out a GABA gene, these cells weren't doing GABA anyway, no one cares. The only set of cells that are actually going to have a functional effect are cells where it's the correct promoter, so it's AGRP, and it's target gene that matters. So in this case, it's got to be AGRP cells that are also GABA cells. And that's the very small set of cells that we're looking at here. It's these AGRP GABA cells in the hypothalamus. There's very few of them, but this technology allows us to specifically target and delete GABA function just from that one small set of cells. So it's not we're going to make a whole brain seizure mouse. We're going to make a mouse that just has the content of GABA function in these cells deleted. And that's really pretty cool. And what these guys saw was, yeah, as it happened, in the experimental mice, they got fatter when you fed them a normal diet, and they got even fatter when you fed them a high-fat diet. So here's the average weight gain on a high-fat diet. And you can see, you know, there's a definite, there's definite loss of body mass, right? So they're using more fuel if you take out this gene. They're getting leaner. The control high-fat diet mice are getting fatter. These mice where we knocked out VGAT are staying thinner. The conclusion is this set of AGRP GABA cells is important for causing hunger, right? It's important for making these mice want to eat more. And if you knock it out, they don't get as hungry, they don't eat as much. They seem to, in fact, burn more fuel, which is kind of cool. And so they stay leaner. They stay at a lower body mass. If you read the paper in detail, you'll see this. And what this is telling you is that these cells don't respond quite as much in the response to ghrelin. Ghrelin is a hunger hormone. It's a signal the body needs to say, hey, you should be hungry right now, go get some food. The wild type guys have more spontaneous activity. They have more of these spontaneous spikes when ghrelin is around. If you knock out that VGAT gene in those AGRP cells, they stop responding to ghrelin. And so the basic conclusion is that GABA signaling in this one small set of cells is a key factor in linking hormonal signals for hunger to food intake. And if you don't have that, you stay leaner. All right, as I said, the key point of that paper is really the methods, the whole Krelok system, and why it allows you to manipulate very specific cell types. If that's clear, great. If it's not, go back, read it again, ask questions, because it's a really important methodological tool, and it's one that's used definitely to this day in many, many different experiments where you want to specifically knock out a certain function in a certain set of cells. All right, 1 second, I'm going to go close the door for my 10-year-old. I'll be right back. We're going to talk about glucose in the brain again. The joys of recording lecture at home. All right, so we've talked a bit about glucose and homeostasis. Good example of adaptation. So hypoglycemia. Hypoglycemia, Latin for too low glucose. So when your blood sugar is too low, and you can see on the screen, if I were to come around somehow and give you all a shot of insulin right now, it would take the glucose out of your blood, it would make you hypoglycemic, and you have to have a whole bunch of responses. Your body is very well set up for detecting, wait, my blood sugar is too low, I better fix this, this is a big deal. So you'd start sweating, you'd feel hungry, your heart would race, you'd have a headache, you'd start getting blurred vision, lots of stuff would happen. Hypoglycemia, your brain gets really mad, there's not enough glucose, this is a big problem. The adaptation comes if you are repeatedly hypoglycemic. And this is really just seen in type 1 diabetes. So type 1 diabetics, you are repeatedly dosing yourself with insulin. On average, that causes about 1 hypoglycemic episode per week. That's clinically actually a big deal. And what happens is you get this thing of adaptation. I mentioned weight gain previously. You gain 10 pounds, your body says, okay, great, that's the new set point. If you are repeatedly hypoglycemic, your brain says, huh, I guess this is normal. Okay, I'll stop responding. And so rather than having this whole set of symptoms, in folks who've been repeatedly hypoglycemic, the next episode of hypoglycemia doesn't cause them. I've had patients at Yale who came into my office. Normal blood glucose is about 100 mg per deciliter. The threshold for going into coma is maybe 35, 37 or something. These guys would come in, we'd check the blood glucose, and they'd be 40 mg per deciliter. They'd have no symptoms. They'd be completely unaware that they were about to go into coma. They were in real danger. And then you ask them, okay, well, what are you going to do? Oh, I guess I should go, maybe I'll go drive home and get a snack. Obviously, that's a really bad idea. They're very likely to go into coma while they're driving. And it turns out that actually the leading cause of death in diabetics under the age of 70 is car accidents. Probably because if you don't have enough glucose in the blood, your brain simply cannot function. We've seen this with the maze task. And so if you have a complex driving scenario where you need to think fast and correctly, you don't have the fuel to do it. So hypoglycemia unawareness is a very negative, but very clear example of adaptation to a new set point, with the brain not responding. This is actually something I've worked on quite a lot. And we can see what changes in the brain. So, antecedent RH. RH is recurrent hypoglycemia. So we have some control rats that didn't get made hypoglycemic. And we have some rats that we made hypoglycemic once a day for 3 days, take their brains out, look at them on the 4th day. And what you see is there's much more GLUT1. So glucose transporter 1 is glucose transporter that gets glucose into the brain. There's much more of that being made in the hypoglycemic brain. Why? This is adaptation. The brain has said, wait, we're repeatedly getting this state of hypoglycemia. I might not get the fuel that I need. I'm going to adapt by making much more glucose transport so that I get the fuel I need. And that works. And actually it makes these animals smarter because the brain is now much better at getting glucose all the time, not even when they're just hypoglycemic, but the whole time. Until you put these animals in a maze, they get really smart. This was completely unexpected. So when I did this work, first of all, it was really cool to get such an unexpected finding. And second, we started thinking, maybe we should all treat ourselves with insulin once a week and we'll get really smart and we can have Dr. McNay's brain insulin clinic and make $1 million. That's great for type 1 diabetics though, because the brain is now really protected against hypoglycemia and can't detect hypoglycemia. It means that in the clinically important case where they're going to get hypoglycemic again and need to be able to respond, they can't because they don't notice they're hypoglycemic. The other reason why we don't have Dr. McNay's range of brain insulin clinics is this. So these are 2 rats, Sprague-Dawley rats is what they look like, white fur, pink eyes, pretty bad eyesight. They're the same age, they're the same sex, they're both male. They've been fed the same diet. You can see there's a big difference in body size. And the reason for that is that this rat has been being made hypoglycemic just once a week for, I think, 12 weeks at this point. And so as part of the brain's adaptation, the brain doesn't just say, okay, I'm going to upregulate glucose transport to myself. It also says, well, if we're going to be starving the whole time, we really can't afford to be spending fuel. So stop. Don't move around. Don't expend energy. Conserve it. And what happens is these rats get massively fat. The body mass is about twice as much, and it's all fat. When we sacrifice these animals into the experiment, we actually go open them up to measure their fat. The fat basically just literally pours out. By the time you're at 12 months, 16 months old, it's kind of like having a rat here with 2 extra rats just of fat kind of strapped. It's really, really quite remarkably gross. It's the fattest rats I've ever seen. We had rats that were weighing 2 kilos, 3 pounds, just kind of flobbing over your hand full of fat. And moreover, these rats don't move. So what you're looking at now is motor activity. And it's a measure taken by a transmitter we put into the belly of these guys, and it moves around giving a radio signal. You can see there's a pretty good diurnal effect, right? So here's daytime, here's early morning, here's late night. Rats are much more active at night. The control animals, they're moving around a certain amount. This is pretty normal. The recurrently hypoglycemic animals, RH, they show that same diurnal effect, but their motor activity overall is about half that of their control counterparts. So they are eating a little more, and especially they're burning much less. Basically, the recurrent hypoglycemic rats are sitting on the couch, eating Cheetos, doing nothing, and becoming markedly obese. So there are several examples here of very clear adaptation because the brain is trying to maintain homeostasis. It's trying to maintain its own brain glucose supply, and it will cause a whole bunch of changes, a whole bunch of adaptations in order to service that goal. Back to obesity. So other than being repeatedly hypoglycemic, which doesn't happen much, what else can make you fat? We saw previously the brain can detect whether foods have calories in or not. Having 0-calorie foods is a well-known risk factor for obesity. And so this is what the paper we saw before, we can get different effects in rats and in humans in brain areas, depending on whether the sweet thing has calories or not, even though you may not consciously be aware of whether you're fat. One other thing that's linked to obesity, viruses, bacteria. Bacteria in particular, both your own endogenous bacteria, but also if you get infected. And so there are viruses that don't appear to cause any major effect, except alter your body mass. We can link these two things together. So one of the effects of having lots of artificial sweeteners is that it changes the microbes in your gut. This is kind of weird. So most of these non-calorific sweeteners don't get metabolized. They do encounter the microbes in your gut, and it seems that they actually have an effect. They change your body's response to glucose, change your body's response to insulin, change how many calories your body absorbs and stores fat. This has actually led to an industry now. So we know that there are certain classes of bacteria that are associated with lean animals versus obese animals. The percentage of Bacteroides compared to firmicutes does seem to be linked to that, and it's causative. If you take that high Bacteroides microbe set from the gut of a lean animal and transplant it into an obese animal, what you see is you can actually reduce the body mass by giving them a lean donor, a bacterial sample from the gut of a lean mouse. And if you look at the body fat, if you give a very fat animal's gut microbiome to a control animal, it increases greatly. There are now clinics where you can go pay to have your own gut bacteria wiped out and replaced using a fecal transplant, which is just unpleasant as it sounds, with the bacteria from a lean individual. It does appear to have some effect. Your body generally, as you would expect, tries to impose homeostasis and gets you back to your bacterial type you started with. So you need repeated treatments, but it does appear to be effective. The microbes in your gut are controlling much of your body mass. If it starts to freak you out that some of your body's functions are under control of cells that you don't think of as yours, here's a much weirder example, right? So this is a carpenter ant. This is a carpenter ant whose jaws have locked onto a particular plant. It's climbed up to the top of this plant. Its jaws have locked onto it. And then the muscle fibers controlling those jaws are being eaten away. Why? Well, this thing coming out of the ant's head is a fungal spore. The fungus has infected the ant, interfered with the ant's nervous system, taken control of it, forced it to climb up and die at a spot where the ant was likely to be eaten, so the fungus could continue its own life cycle. This is a complete hijacking of the animal's nervous system by a fungus. Just recently, an anti-zombie fungus was discovered. So a second fungus that infects ants and lives in a symbiotic relationship, protecting them from being infected by this zombifying fungus. All right, this is really cool stuff. Parasites in general are utterly cool. And we'll come back to this when we start talking about yourself and what makes you you. Because as you might imagine, there are classes of bacteria and microbes that can actually, you might say, infect your body and change your personality. And so the question of who's in control in your brain gets kind of complex. We'll come back to that. Let's go back to food control for now. Lack of sleep. Lack of sleep is well associated with gaining body mass. This has been tried to explain several ways. The most simple and apparently correct explanation is mostly that when you stay up late, you get hungry and you eat more. We're not going to talk about sleep much this semester. I think it's going to be one of the things we miss. But in fact, one of the ways that you become sleepy is by your brain sensing a low fuel state. And so if you have a snack, that can actually put off sleepiness for a while. But definitely staying up late is associated with high carbohydrate snacks and therefore body mass gain. Pollution. There are lots of things in the environment these days, pesticides, fertilizers, which can interfere with your body's control systems and cause you to gain weight. Then there are some genetic causes, right? Prader-Willi syndrome. This is the most common genetically identified disorder regulating body mass. About 1 in 12,000 people. Causes hyperphagia, that is massive overeating and constant hunger. So basically these folks are never satisfied, they're never satiated, they're always starving. It appears to be a deficit in the growth hormone ghrelin system, so that hunger hormone that turns on, turns off hunger. Notice that even here, the body of these individuals is in fact trying to impose homeostasis. So this guy weighs, I forget, I think it's 700 pounds. He's taking in about 5,000 calories a day, sometimes 7,000. That's 1 to 1.5 extra pounds worth of fuel every day. And so if all of that were being actually stored, he'd be gaining 500 pounds a year. You know, that's much more than actually he gains. Obviously, he's massively fat, but in fact, his body is trying to do its best to maintain a body mass and not store all that extra fuel, even though it's a losing battle. So Prader-Willi syndrome, single gene disorder. Obviously, there's an interaction between genotype and phenotype, between your genes and the body mass, in this case, you display. So this is Puma Indians. You know, there's a migration of those Puma Indians. They came over a land bridge down through Alaska, migrated down through Canada, now are living both in Mexico and in New Mexico. And as you might perhaps guess, the food supply is very different in central Mexico than it is in New Mexico. And what you see on the screen is that the percentage of folks with type 2 diabetes is much, much higher in New Mexico. Even though genetically identical populations, you know, both have, at least in theory, some food availability, but the over-availability of food in New Mexico is causing this really marked increase in obesity, marked increase in type 2 diabetes. The genotype of this population is especially thrifty, is especially good at storing fuel, and honestly, in largely part, it reflects their evolutionary history, where marching and migrating down through Alaska and Canada, calories were not plentiful. Now when they are, the brain is gorging on them because the brain's still assuming, yeah, tomorrow we starve. I've talked a couple of times here about this concept of the brain being selfish, as though it's not part of you, as though you're somehow separate from your brain. You can hold on to that thought. You can also definitely realize that in some cases, different parts of your brain are arguing with one another. So when we were looking at sensory function and so on, we had a whole bunch of senses being integrated in the parietal lobe. That information was going over to the frontal lobe to make plans. And often the role of the frontal lobe is to act as a brake, is to put a brake on some of the plans you might otherwise carry out. Somebody's really being annoying, default response might be, you know, punch them in the nose. Your frontal lobe says, no, bad idea, you know, don't overreact, you might get arrested, they might hit you back, this is all a bad plan. In the context of food, in general, your limbic system, your hypothalamus, have all set up to say, food, eat, more calories, chocolate, cookies, yes. And your frontal lobe is often part saying, no, you'll get fat, you really have already had 17 pieces of cake, you don't need another piece of cake, stop that, get control of yourself. Self-control is a key, you know, concept here. In some cases, that can go overboard. And so eating disorders where you don't have the ability to accurately regulate and accurately control your food intake. Anorexia, right? So anorexia is a condition where the patient perceives themselves as being overweight and tends to refuse to eat what would actually be a healthy amount. If you show them a piece of tasty looking pizza, normal subjects, the amygdala here at the back of the brain is lighting up saying, yeah, looks good. Let's taste it. Sounds like a good plan. That's much less active in the patients with anorexia. And instead, what you also see in those patients is greatly increased activity in the frontal lobe. And basically that part of the brain is saying, if you eat that, you'll get fat. You don't want to do that. Bad, bad. Stop being happy about this. That self-control is a real brain function, right? And it's actually kind of interesting to examine what causes you to have self-control, what uses it up. There's a great experiment where students are recruited and they're taken into one room and asked to memorize a number and then told, okay, great, hold that number in your head, walk down the corridor and tell it to my colleague in the other room who's waiting to see how good your memory is. So it's presented as a test of memory. And there are actually two groups here. One group is given the number 49. Remember the number 49, go tell my colleague. That's a pretty easy task. The second group is given the task, okay, remember the number 17,49,269,342,073. Okay? Try to remember that as best you can. Great. Go tell my colleague. On their way down the corridor, both groups are accidentally interrupted by a food tester who's doing a different, the shtick is they have two types of food they're thinking about putting in the student cafeteria. Would you like a free sample? And they're offering you a free sample of a chocolate cake or a carrot snack. What you see is that the people who are given this ludicrously complex number tend to go in far more numbers than you would expect for the chocolate cake. The folks who are told 49 are really quite likely to have the carrot. It looks as though that act of memory, the act is using up glucose and using up brain reserves to hold this number in memory, reduces the brain's ability to exercise self-control. So we think of self-control as being just another function that really requires brain fuel supply to actually allow you to exert self-control. And so the frontal lobe is burning glucose to tell you to stop, to act as a brake. Here's a good example. Okay, this is the Stroop test. Read these words, except don't read the word. Tell me what color the word is printed in. So here, for instance, I'm going to do it for myself. What you would say is red, blue, green, yellow, green, black, blue, red, yellow. And as you do that, you actively inhibit reading the word itself. Your default response to any word is to read it. You can't stop it. You can't prevent your brain from doing it. You're going to read the word. And so when you're told, just tell me what color it's printed in, you have to say red instead of purple. That requires you to actively inhibit. It turns out that we can show that very effectively in other ways. This video isn't going to show. I'll show it to you next time. We'll skip it for now. If you give people that Stroop test, right, and you measure how many errors they make, and just before that, you've given them a test of self-control. Sit here and don't do something that you would normally by default do. If they've had to do that task, they will normally make much more errors. If you give them a sugar drink, though, now they will make fewer errors. They will come back to baseline on this task. If you give them a drink that doesn't have any calories in, they will continue to make lots of errors. And so it looks as though glucose, unsurprisingly perhaps, as well as fueling things like memory, is also what you need to exercise willpower. You guys are probably aware of the fact that if you've been thinking really hard or you're really tired, you are less able to resist the cookie or make sure you make good choices. All of these different functions in your brain are all using glucose. And so it's a single common fuel that we actually think of as being, in some cases, what we think of as self-control. It's glucose. Think of as brain power, ability to think well. That's glucose. Your brain's fuel supply is critical, all this stuff. Back to eating disorders for a second. Anorexia is a pretty dangerous disease. Kills about 10% of patients. Bulimia is similar, but tends to involve consuming food first and then purging it, often by vomiting from the body. rather than not eating in the first place. Similar symptoms in both cases definitely causes a distorted body image. People really do misperceive how they look. And there's definitely a social component. So it most often affects young, female, fairly affluent and well-educated young women. They appear to have this societal impression they should look different to the way I do. So this is Kate Moss. If society is telling you this is how you look if you want to look good, and you look in the mirror and what you see is this, even though this is how you actually are, there is a big impetus to not eat, to try to get thinner, because that's what society is saying you have to do to look pretty. So there's a huge societal component to anorexia. I told you that people misperceive themselves. That's absolutely true. If you give people a set of pictures of different body mass types, you ask them two things. Where are you? And what do you find most attractive? Non-anorexic, most folks who are not suffering from any eating disorder can accurately place themselves, and they'll say, this body type in the middle is the most attractive. Patients with anorexia universally say, I'm over here, and this is what is attractive. This is how people should look. This is what looks good. So they are bad at perceiving themselves, and they are bad at perceiving what a correct body type should be. There's a really widespread set of issues in the anorexic brain. How does anorexia develop? Turns out there's an interaction. Like many disorders of mental function, you require both a brain that is predisposed, that has a setup that allows for the miswiring, for the misfunctioning, and then you also need a trigger. And often for anorexia, that can be puberty. So some combination of hormones, stress, like I said, cultural factors, watching Kate Moss play down a catwalk. Something in there interacts with a brain that is set up to be potentially anorexic, and you get into this dieting cycle. Many people will recover quite happily, but about a third to a fifth of folks get into this really nasty cycle where the weight loss causes further changes in the brain, which further exacerbates some of these negative brain traits. And now you're in a cycle that it's really hard to break out of. Notice that you don't just want to grab these folks and give them mass amounts of food. That would actually make them feel bad, which would cause a negative memory association with the food, and so actually make things worse. They'd be even more likely to avoid food. Oddly enough, if you've got folks who really have been literally starving and you feed them a full big meal, they can develop anorexia because the consequences of that rapid overfeeding are too great. And so you really want to just try to give small, frequent meals and have positive, happy associations with the food, nothing negative. In the brain, now what is that predisposition I'm talking about? It has to do with the balance of serotonin receptors. So we saw these way at the start of the semester in the context of anxiety. The balance of different serotonin receptor types, whether it's 2As or 1As, is also really important in terms of regulating things like, food consumption. Are you able to have normal levels of happiness? Do you have dysphoria, which is an inability to have regular amounts of pleasure? So if you have too much of the 1A receptor, which is this negative, controlling receptor, that can trick you and push you into this dieting cycle. As you lose weight, what that tends to do is cause you to reduce serotonin production, and that can actually cause more of these 1A receptors to get made. This is the brain's, response to homeostasis. If you're getting less serotonin released, make more receptors, but that can increase your dysphoria, can increase your lack of pleasure, and unfortunately trigger you back into this whole nasty dieting cycle. So balance of serotonin receptors is one key thing. I mentioned previously, pleasure is really important, right? We don't just eat to get food calories, we eat for pleasure. And so if you're not getting the regular amount of pleasure from food, or in particular, if food has become an object of negative emotion, of disgust, that can really drive you into a state of anorexia. If you look at the amount of dopamine binding in recovering anorexics and control people, you see far more dopamine binding in the recovering anorexics. What you see then is this massive increase in the brain that someone who's been through this and is now adapted out or managed to cure themselves But notice it's far more. It's not just back to baseline, it's far more. It's possible that one of the problems here is that a small amount of food is enough to trigger the right level of pleasure, to trigger the right level of dopamine binding. Whereas you don't need, one donut, like the control brain. Maybe you need 1/10 of a donut. And more than that feels like it's too much, like it's just overindulgence. And so just like obesity, where getting too little pleasure from a donut might be a trigger for obesity, getting too much dopamine from a donut might be a trigger for something like anorexia. Yeah. This is certainly true in dieting. If you go through the pain of trying to reduce your food intake, your brain says, no, homeostasis is good. I want to keep my body mass, and I'm going to change things so that it feels bad for you to diet and feels extra pleasurable for you now to go to have a donut. And so you can actually get rebound weight gain that takes you past where you started. Excuse me. Last bit on the abnormal setup in the anorexic brain. And again, it has to do with this balance between different brain regions. In the healthy brain, here's a nice looking piece of cake, activates your amygdala, says, hey, looks good. Prefrontal cortex, not particularly active. All the smell and taste signals are positive. Overall, the brain's going to say, great, that looks good. And your behavioral response should be to go eat some food. Yum, yum, yum. Short-term positive effect. In the anorexic brain, that amygdala may still get activated, but it also causes overactivation of the frontal cortex, this brake system. And so that brake, this top-down negative, no, bad, that's going to make you fat, tilts the balance of how you respond in favor of avoiding the food. And unfortunately, what that means is you're going to have a memory that seeing food made you feel bad, and so you're more likely to avoid in the future. You get this horrible spiral. And so there's an overbalance towards long-term consequences, staying thin at all costs, out of whack with the short-term, have some pleasure, eat some cake, it's going to taste good. We've reached the end for today. Hope you guys are staying safe. Hope you guys have, the food, the toilet paper, all the stuff you need. I really do want to help with not just class stuff, but if there's any other thing that's causing trouble, if you need assistance from the university, someone to talk to, whatever I can do, I really genuinely am here and available and more than willing to do whatever I can to help. So I'm going to send this on the video system. It should be up on the YouTube channel. It should be available for your viewing pleasure. I hope it was comprehensible. I hope you're enjoying the videos. Give me feedback if you'd like, and I'll see you on Tuesday. Bye, guys.